I'd just like to tell you about a program that we've had in train for quite some time, and which I have taken over as the director of only in the past 12 to 15 months, looking at post-exposure therapies for this particular virus, VEEV virus, as we call it. Whoops, hit the wrong button there. VEEV is, like most viruses of concern, is an RNA virus. It's got a positive strand genome, and all of its structural proteins are translated as one large polyprotein and then proteolytically processed. It's responsible for large outbreaks in human populations in the Americas. But our interest in it, our interest in it is because although it's normally transmitted by by mosquitoes, it is also readily transmissible artificially by aerosols. It has a very low infectious dose, and it has a history of weaponization in the United States back before the Biological and Toxins Weapons Convention was signed in the 60s. There are also no licensed vaccines or antivirals for the virus. There have been a number of studies over the years looking at antibody therapies for VEEV, and a number of monoclonal antibodies have been generated. One of the things that has become apparent is that antibodies to the E2 surface glycoprotein, which is found on the surface of the virus and on the surface of the infected cell, this can generate neutralizing antibodies far more readily than the larger E1 protein. And these antibodies will prevent the growth of the virus in tissue culture. They'll recruit downstream immune mechanisms, and they have the potential to ameliorate disease. So if we look at the ability of these antibodies to prevent disease, our team took a whole panel of antibodies, most of which were not generated by us, and looked at them in vivo. And if you give the antibody, typically for this experiment, time 0 is when we would challenge the mice. We would give them antibody either 24 hours prior, immediately post-challenge, or at various times previously. And we found you could get a level of post-exposure therapy. You could get a level of protection, and this differs from antibody to antibody, but clearly demonstrates that there is the potential of these antibodies as a post-exposure therapy. That time scale there is in hours. Okay, sorry. And we took this data to the modelers at Dstl to allow them to either justify or not justify for us the potential for going on and developing a medical countermeasure. When we look at these studies here, we know these animals are infected because we directly infected them. In a real-life scenario, on a battlefield where someone releases an aerosol, you don't really know how many people are going to be actually exposed, physically going to breathe in the virus. Obviously, the meteorology, the weather conditions will have a significant role to play in the viability of an aerosol and so on and so forth. And so what we wanted the modelers to say is, what was their prediction for what would happen if we had a medical post-exposure therapy of the sort of level that we could get from these antibodies? And what they told us was that without a therapy, the probability of infection of our troops was very high. If we could get a therapy on board within 2 hours, then the probability was very, very low, and as time went by, that probability increased. And you can see quite large error bars there. But this was certainly a justification for us to go on. And the question then for us was which monoclonal antibody to look at. Now this table merely tells you, you don't have to look at it in great detail, all it's really telling you is that there are an awful lot of strains of Venezuelan Equine Encephalitis Virus. Some of the monoclonal antibodies recognize one, some of them recognize a few, and so on and so forth. And when we started this study, we didn't know how many of them recognized how many. Most of them were raised to the Trinidad donkey strain. And if you see here, the strain FE37 and Mocambo, these were originally VEEV viruses, but they've subsequently been reclassified as their own species of virus. Everglades in this case and Mocambo in that case. So we needed to look at the panel of monoclonal antibodies we had and see which ones would recognize a multiplicity of strains. And this is just two of the antibodies we've examined for comparison purposes. You can see here 1A3B7 recognizes a great number of strains, including Everglades and Mocambo, which are actually related species rather than strains. And this is just a representative of the other antibodies we tested, which you can see will drop out against certain strains. So 1A3B7 was the one for us to look at. This is, I should say, that this is just recognition of the purified protein. If we look at this table, we're actually looking at protection with 1A3B7 against various strains. And these are given 100 PFU, which is a very large challenge with VEV. In fact, in mice, in biopsy mice, less than 1 PFU is enough to give a lethal dose. And that's all to do with particle PFU ratios. So this is quite a large dose, and as you can see, the antibody given pre-exposure in this case is protective against that multiplicity of strains. We get on to, perhaps for the purposes of this conference, the meat of this particular presentation, in that this antibody is a murine antibody, and that's fine when you're testing it in a murine model, but in the final analysis, we want to put this into people. And as you're all aware, the historical approaches to desensitizing murine antibodies was initially to make a chimeric antibody where you replace the murine constant domains with human. You still get an immune response, and so you go along and you actually remove the framework regions from your murine antibody, the CDRs, and put them into human frameworks to give you a humanized antibody. That should address the issues of clearance of antibody after its administration, and also reduce the probability of adverse reactions. And I think I've probably just gone through that, so we don't need it, and we're all preaching to the choir anyway. To actually identify the CDRs and framework regions for this antibody, we went to some collaborators at UCL who ran a series of algorithms, because there's always a little bit of play with where the CDR ends and the framework begins. And we came up with a number of different molecules for the light chain with slightly different ends for the CDRs. And these eventually were turned L6, A26, and B1. And we did a similar thing for the heavy chain. So we ended up with 3 light chain molecules and 3 heavy chain molecules, which we mixed and matched together to give us a combination, 9 different combinations. And when we looked at the ability of those combinations to bind to the purified antigen and also to inactivated VEV, we found only 1 of them would bind. The other 8 were completely negative. So in some respects, that's not surprising. They all had slightly different CDRs, and in the other respect, you only need 1 for this kind of study. So we took that one, and we looked at its affinity. The comparison here is with the chimeric antibody, which we'd also made earlier on, which has human constant regions, but the murine variable regions, and which has the same affinity as the parent murine antibody. And as you can see, these two are essentially giving us the same affinity, which was good. We then took the humanized antibody. Sometimes you'll see it referred to as H1A3B7, sometimes as Hu1A3B7, and looked at it against the multiplicity of strains, including Everglades and Mucambo, to check that it still recognized all of those strains. We had no doubt that it would, but it obviously needed to be done. We then looked at the ability of the antibody to neutralize virus. This is the parental murine antibody in the dark blue versus the humanized variant, and against the type strain of VEEV and against Everglades and Mucambo, you can see it still neutralizes the viruses to the same extent. Then we went on to our pharmacokinetic studies, in which we put the murine parent into mice and then took blood at various intervals to determine the half-life of the antibody in the mice, and the same with the humanized variant. And as you can see, the rate of decay of the humanized variant is much greater, to the extent that the murine antibody, the parent, has a half-life of about 22 days, and the humanized variant a half-life of about 7 days. But this is the reciprocal experiment to what we would want to see in people. We would expect the murine parent to have the longer half-life in mice, and we would expect the humanized variant to have the shorter half-life in mice. We'd expect these values to be flipped if you were to put these two antibodies into people. And this was an indication that what we were seeing was what we would expect to see. So if I now just run very briefly through the mouse challenge model, we can challenge mice intraperitoneally or subcutaneously or by aerosol with VEEV. And most of the data I'm going to show you will be with aerosol, because that's what we're actually interested, that's the indication we would want to license it for. All this is really showing you is that the models, the mouse model is very similar to what we know of the human disease, with the difference that in the mouse, it's very easy to get 100% lethality, whereas in the human, which is mostly natural infections, the case fatality is actually very low. That's not really a problem. It's very difficult to get anything licensed on a mouse model that has 1 to 5% case fatality. So we're quite happy to have that particular difference. But everywhere else, it's largely the same. So if we look at protection with the humanized antibody now against, not the mouse parent, against lethal challenge, this is an injected challenge. It's an intraperitoneal challenge, and the antibody is given post-exposure. But what we're really trying to do here is to see how much antibody we need. The antibody isn't given post-exposure, it's given pre-exposure, sorry. What we're really trying to do here is see how much antibody will protect. So given 24 hours pre-exposure, you can see this is the standard dose that we were thinking of doing, but even if we drop it by almost an order of magnitude, we're still getting a very high level of protection.
So what do you inject that?
In that experiment, this experiment, they would both have gone intraperitoneally. Standard route for administration of the antibody in these experiments is intraperitoneally. This is onto an aerosol experiment, an aerosol challenge. It's with the MENA2 strain of VEEV. We've also done this with the type Trinidad donkey strain. And we're giving, I think we gave 300 PFU of virus in this experiment. As you can see, if we give, ooh, wrong button again. If we give a control human IgG, we don't get protection. Although we've given a very large dose of MENA2, we haven't most of our studies to determine the actual median lethal dose have been done with Trinidad donkey, and in all probability MENA2 is a little bit less virulent in mice, but still we're getting a 90% kill. But where we use the humanized antibody given 24 hours post-exposure, we're getting 90% survival, and where we give it 48 hours post-exposure, we're still getting 80% survival. So in a lethal challenge model that takes only between 5 and 8 days to run its course, to be able to give your medical countermeasure 2 days post-exposure and still get 80% survival is pretty good. If we look at the Mocambo virus, we get a very similar story. Here you can see that with the control human IgG, we're getting 100% kill, but with both 24 and 48 hour post-challenge administration of the humanized antibody, we're getting complete protection. If we go on to Everglades virus, again, the same story. The control human IgG, all the mice are dead by day 8, and whether 24 hours or 48 hours post-challenge, all of these animals are surviving. And that, for us, was as good as we really could have expected it to be. If we go on now, there we have efficacy, and we need to go on to the potential for adverse events. I don't know how I'm doing for time, Jeremy, because I haven't been watching. Okay. We were obviously concerned about the immunogenicity of this humanized antibody, and we actually came to ProImmune and asked them if they would be able to help us out with this. And much of the next couple of slides you'll have gleaned actually from Jeremy's earlier presentation. What ProImmune did for us was to undertake a T-cell proliferation assay with naive donors. The cells they used were CD8 depleted, a minimum of 40 donors representative of the global human HLA class II profile. Proliferation of the CD4 T cells is measured by the decrease in fluorescence because they're preloaded with the carboxyfluorescein dye. So in each cell division, each daughter cell gets approximately half of the original dye, and you can see then that fluorescence signal depleting. The controls included purified protein derivative of Mycobacterium tuberculosis for a memory response and keyhole lymphocyte hemocyanin to look to control for naive responses. So this is basically what we would expect to see, what we would hope to see. Along here on the bottom axis, I think we have, on the vertical axis we have CD4, which is just staining our CD4 positive cells. So these should all be up here. And then on this axis, we have the carboxyfluorescein. And if the signal up here in our CD4 positive cells, double positives, should move downwards, move to the left, then that would indicate that we've got, that we've stimulated a response, which you would see here. That allows us to get a cell division index, which is a measure of proliferation. If we look at our chimeric variant of this antibody, so it's got the human constant regions, but it has an intact mouse variable region. You can see here with the light chain, and again, each of these colored bars represents one donor. Each of these numbers represents a peptide, and these were 15 peptides overlapping by 3 amino acids. These are the controls of PPD and KLH. And you can see that we are getting a fair number of donors responding to a fair number of peptides. With the heavy chain, again, a fair number of donors responding to a fair number of peptides, but then we have this sort of clustering in this region. So this chimeric antibody really is quite immunogenic. But if we then go on and look at the parallel experiments with the humanized antibody, we can see that by and large this response has gone. And to be honest, I didn't realize quite how clean this was until Jeremy showed us the Humorics data, where you were getting a response to to 4 peptides. So this really is quite clean. It's very nice. We were delighted about it and sang about it to our bosses, our paymasters, in very loud voices. That's where we are. And if I go now through future work, we have to look at production of antibody because our yields in this are very low. This actually I should have deleted because that was the data I showed you on the post-exposure therapy. So that's actually done. We do need to understand the protection offered at different times of administration relative to the level of challenge. And that's all work that would need to be done in a future study and further assess suitability for humans. But we are waiting for our paymasters. We're being filmed, aren't we? We are waiting for our customers to decide whether or not they're going to transition that into a further development program. But we certainly hope that they will, as a 2 days post-challenge immunotherapy is certainly very good in that kind of truncated model. So in summary, we have a monoclonal antibody that's effective in treating VEEV. It's derived from a mouse and therefore may cause adverse reactions in humans. And we produced a panel of humanized antibodies, one of which retains biological activity and offers protection against lethal challenge, post-exposure protection in a small animal model of the disease. And it therefore has the potential to be a useful therapy. And that's not just for military use, but also in the periodic outbreaks, zoonotic outbreaks that you see in people. And of course, if we were unlucky enough to have a laboratory accident. And then just our acknowledgements to our various collaborators, ProImmune and UCL and the guys at Wyeth. And I should also mention John Rurig, who made the vast majority of the antibodies that we've worked with originally, the murine versions. Thank you.